Right, so let's talk about two types of variables right now. Once you do an experiment, you're going to need to make sure that you identify your independent variable and your dependent variable. So the factor that is changed is known as the independent variable. And so that is the thing that you're going to be changing throughout the experiment, and that is going to affect the dependent variable. So the factor that is measured or observed is called the dependent variable. Right. So remember, independent variable is the thing that you change throughout the experiment, and that affects the dependent variable. So an example of this, right? Because that may not make a lot of sense right now, but when you think of this example right here, then growing these plants, there's an experiment, right? In the experiment, the independent variable that is changing throughout is the amount of water that you give a plant, right? So this is going to change. So let's say this first one right here, you're only giving it about 5 centimeters of water a week, or let's say 5 milliliters of water a week. This one you're giving 10 milliliters of water a week, and this one 15 milliliters of water a week, right? So that you're changing that throughout the week. And so every time you do that, it is changing the variable, right? So 5 milliliters, 10 milliliters, and then 15 milliliters, and it's changing, right? And that, like I was saying, the independent variable affects the dependent variable, right? So it may be that the plant that you, depending on the type of amount of water you're giving the plant, then the plant will either grow more or it'll grow less, right? So the independent variable is the amount of water, and the dependent variable is just the size of the plant, right? So again, independent variable would be 5 milliliters of water for one plant, 10 for the other, and then 15 milliliters of water for the other. And then the dependent variable would just be the size of the plant, right? So you would just measure this one is about 5 centimeters, this one is about 10 centimeters, and this one is about 20 centimeters. So that is a dependent variable for this experiment, right? So those are the independent and dependent variables. Next, you have a control variable. So the control variable in an experiment, it's just this is when the experimenter makes a special effort to keep all the other factors the same. Remember, we were talking about how you only want to test the one thing and you want to try to keep everything else the same so that you're only focusing on the one thing. If we're talking about the plants, we want to make sure everything is the same. The soil is the same, the amount of sunlight, the type of water. The only thing that we want different is the amount of water for each one, right? So that they will not affect the outcome. Those factors are called control variables. So in this case right here, a control variable would look something like this. Here you see that we have the control and the experiment. In the control, it's pretty much everything that the experiment is getting, except here you see they are using fertilizer in the experiment. Whereas you see over here, there is no fertilizer. So again, they're trying to control for everything, and so they're controlling for everything except for the one thing that they're trying to test, which is the fertilizer, right? So the fertilizer is what is they're experimenting, see which one is more effective for plant growth. And so, but you need to compare it against something, right? Because you need to know how different is this fertilizer compared to like not using fertilizer. So you use a control group so that you can compare it to later, right? Sometimes when they're testing a drug, they will give one group of people the actual drug, and then they'll give the other group of people just like a fake drug, a placebo, a sugar pill, or something like that, so that they can compare one group to the other. Right. And again, as I mentioned, that the controls are, you're not testing them, you're just using them to compare, right? So again, you do one group that does get the medication and the other one that doesn't get the medication. Because if you do these experiments and you see that the group that got the medication has the same results as the one that did not get the medication, chances are your medication doesn't work, right? But if the cholesterol or whatever the drug was doing is a lot more effective, and like 80% of the people that got the drug are doing a lot better than the other part that did not get the drug, then that's a good it's good to have the control because you can make that comparison. Right, so let's look at an example, something that can happen like in just everyday life, right? Doesn't necessarily have to be in a science lab that you use a scientific method. So the example here is, suppose you want to figure out the fastest route to walk home from school, right? So you're going to try several different routes and time how long it takes to for you to get home by each one. Since you are only interested in finding a route that is fastest for you, you will do the walking yourself. Right? So what are the variables in your experiment? Varying the route is the independent variable. That is again going to be the thing that you're changing each time. So like in the example we saw earlier, the amount of water that you're giving the plant, in this case, it is the route that you're taking, which is changing every time you're doing the trial, right? So the what you're measuring though is the time it takes to walk those routes, right? That is going to be the dependent variable. So in the plant experiment, the dependent variable was the size of the plant, right? We were measuring the size of the plant. In this one, we are actually measuring or taking into account the time that it takes to walk each route, right? So just like the way the water affected how much each plant would grow, the route here is going to affect the amount of time it takes to do each one, right? So keeping the same walker throughout makes a walker a control variable. And think about that. This is very important, right? You would want to have the same walker to do all of these trials because you don't know, or to walk all these routes. Because if you use different walkers, maybe one of them is like the super uh, Olympic athlete, right? And the other one is just like this really lazy couch potato. And the other one's just like your average person. And so each one is going to have a different speed, but it's not going to be based off of the route. It's going to be based off of their like athletic ability, right? So that's why you want the same person to take every single route so that you are limiting those things, right? You're just focusing on the one thing that you're trying to test, which in this case is which route is faster, right? So which is just going to take the shortest amount of time. So that's why you want to use the same walker for each one. Right. And then you, for this one, you could do like little data collection, which you would get the results of the experiment and you put them on graphs or tables or things like that. It's usually better in graphs and tables because people can understand that better than if they have to read like paragraphs and paragraphs worth of writing, right? And then you can have quantitative data, which has to do with numbers. Remember, quantitative is number related, or you could have qualitative data, which is not number related. In the case of the walking, though, it would probably be quantitative because we would have numbers for the time for each of the three routes that the person took. Right. And then after you do all that, you get all the information, tables, graphs, you read through it, then you make your conclusion. Right. So in the case of the plant, you would just choose which plant grew the tallest, and you would say that this is the best amount of water for the plant. When it came to the routes that you were walking, the conclusion would be like whichever one had the shortest amount of time, that would be the best route to take. Right. And you would get all that from the data that you collected during your experiment. So the conclusion should clearly state your final results and whether or not the hypothesis was supported, right? So if you were doing the walking, the different routes experiment, you would hypothesize, I think route A is going to be the fastest. And then when you do that and you find out, hey, route C was the fastest, then you would say my hypothesis was not supported because I hypothesized that A would be the fastest, but then it turned out to be C. Right. Same thing with the plant. You could have hypothesized the one that got the most water was going to grow the tallest, but maybe it didn't, right? Maybe that was too much water for the plant. So your hypothesis would either be supported or not supported. In the case of the plant, it was not supported, right? Because you thought one that got the most was going to grow the tallest, but it ended up not being the case. And so, in order to verify the results, you have to retest because you can't take anyone's word for it. The person that linked autism to vaccinations, they spread that wide and far, and a lot of people believe that, but no one else was able to replicate the same thing that he was able to. And so, it just his conclusion was false. Like it's not true, but everyone believed it. But once people started retesting it, no one came up with the same conclusion that he did, and so he was actually like disgraced and barred from the the place where he was studying science at. Right. So remember, just a recap right here. Solving a problem, problem solving, and scientific method are just one and the same. Right. You got to identify the problem first, and then you state the observations about the problem. Right. Gather a lot of information about it. Then you form a hypothesis, which is just an inference based on all of the information that you got, all of the data, all of the evidence. And then you design an experiment to test the hypothesis. You collect the data, form a conclusion, and then retest it to make sure that it's going to be consistent every time you do it.